Great. So it's my pleasure to introduce Olivera Grujic today, who's defending her PhD thesis. She's been here for, she's been for UCLA actually as long as I have. She actually she came to the department 2007, and then she took some time off, and she's kind of she's now finishing. Great accomplishment. Most recently, she's been working in Jason Ernst's lab, and she'll be talking about some of the projects that she did there. In addition to being a great student, she's also a real staple of the community in the department, and has been always very helpful with just recruiting and just helping out with a lot of things in the part of the community, and as evidenced by how many friends she has here. So it's a great pleasure to introduce Olivera for her talk.
Thank you. Thank you all for coming. Yes, I was, since I've been here so long, most people that started with me graduated. And so I did not expect the big turnout, so thank you very much for coming, my future colleagues. Yeah, so like Professor Eskin said, I had a long journey here at UCLA, and that's reflected in an unusual path of having 3 different projects with several different advisors. And so I will focus my talk today on my latest and greatest project for the most part, but I feel that I do need to honor the other 2 projects as well, and so I will spend some time on them as well. They're not in chronological order, they're actually in order of large data, because this is computer science thesis, and my mission when I started this PhD program was to use computers to help people. And bioinformatics was an emerging field at the time. Actually, bioinformatics department didn't even exist. So I started working on what used to be a large data at that time, which is the middle project, which is the gene expression data. And then I went back to another lab and worked on something that was actually older, and that's very small data that is not measured in terabytes or gigabytes, but rather in just a few sample points. But it has interesting characteristics about dynamics. So when there were no big sequencing machines and so on, people had very little data, but they were using functions to demonstrate dynamics. And now we have far less dynamics, but we have a lot of data. So that I will mention in the end. The large data in the middle, and the latest and greatest is the large, huge data that is happening right now. And so first, I am going to talk about the method that I developed with Professor Ernst, and it's called Predictor and Identifier of Conserved Elements. And I call it Pickle. It has a symbol for me as Pickle, but I don't think he's going to let me publish it under that name. But so, I'd like to start my talk with a pie chart, and I promise I will have a bar chart in my slide deck as well. And that goes with one of those things I learned in this long grad school journey, is that you can make a pie chart of bars and bar chart of pies. Like pies being apple pie, pecan pie, and so on. And so this is not a pie chart of bars, even though it can kind of represent the bars in Westwood, given that 2 out of 3 major ones closed while I was actually during my tenure here, right? But it's representing something different, and so it's a little bit difficult for me to give this talk. It's a computer science thesis, but I have a lot of biologists and bioinformaticians in the room, so I do have to go into some very rudimentary details for some of you, and at the same time, I do need to get into some specific computer science details that some of you might not find as interesting. So please bear with me if something is too simple. there's probably people in the room that have never heard of it. So at the time I started at UCLA, human genome was sequenced, and the dogma of molecular biology was saying that one gene codes one protein. And so you have this long strand of DNA, double-stranded DNA, and breaking things. And so on the DNA there are genes, and genes code proteins. So this is the double helix, so double-stranded DNA. So we call basically these strands base pairs because they're two strands. And so they're called base pairs or nucleotides, and in computer science terms, they're called letters. They're 4 letters, A, C, T, and G, okay, for these nucleotides. And so each one of these base pairs, 3 of them code for one amino acid. It's very simple, computer science-wise. 3 different letters code for a certain amino acid, different 3 letters code for a different one, and then amino acids, the chain of amino acids is a protein, so basically genes code for proteins. And so we have 3 billion base pairs of those. So the simple thing would be, well, we have enough, and that codes for proteins, and we know how genome works. But turns out it's not that simple. Turns out that only 1.5% of these 3 billion base pairs actually code for proteins. And we call them exonic or protein coding region. Actually, now the term is 4% is exonic. And when I started at UCLA, the rest of the genome was considered junk DNA. What a scientific term, junk. So now we're changing that non-scientific junk term into an actual term called functional genomics or functional annotations. Functional, again, there's this word function, but it's not the computer science function where X maps to Y. It is actually biological function. So recently, with the new sequencing technologies, there have been studies showing that this is not junk. It's now called exonic, non-exonic, and it actually has a function. And it might have different biological function, and it might have different functions based on in different cell types or in different tissue types and so on. And so how do we know that? Also there were these, during the past decade, these GWAS studies that were attempting to find the variance between the individuals and trying to identify the ones that are causal for a particular trait or for a particular disease. And then it turned out that even that is not as simple because these variants were linked together. And so it's hard to tell which one is causal among them. So the problem is, it's called that they are in linkage disequilibrium. So even though we have a lot of data from GWAS studies, we're still not at the point in science where we can tell which single nucleotides are important, or SNPs, we call them SNPs, right, which variants are. And so the mission right now of science and contribution of this project as well is to help to identify in this 96% of the genome what are non-exonic genome, what are some important locations. And so that 96% out of 3 billion base pairs, that's a really big number still. Okay? And so a little more introduction just so that you can understand the rest of the talk is about this non-exonic gene annotation, so non-coding DNA. So this is, the X is a chromosome, and the chromosome is made out of a material called chromatin. So you hear me say chromatin a lot. And so chromatin contains DNA. And then what we found out is that the DNA is actually wrapped around the histone proteins. So when it's wrapped around the histone proteins, and there's like a nice picture here, it's like an octamer, so it has 4 different types of molecules and twice of each of these histone proteins, H2, H2, BH3, and H4, but also hundreds of other histone proteins. So when it's wound, when the chromatin is wound around them, it's inactive. And if the genes are on this portion, nothing happens, right? So what we are interested is when the areas of open chromatin, so when it's unwound from the histones. And so maybe in this picture on the bottom, we can see that a little better. So the green area, I don't even know how to use this. The green area is the gene, right? But then there are all these other elements, regulatory elements. And so some of them call enhancers, like gene enhancers, gene repressors, gene silencers, gene promoters, and so on. And then one more interesting one is transcription factor binding sites. And so what's interesting about that one is that the transcription factor binding sites are again located in the area of open chromatin. And so we focus on that, again, area of open chromatin where the transcription factor can bind. There's also other epigenomic areas that I will talk less, but I'll just mention. There is some non-coding RNA, and then there's also methylation and acetylation, so these molecules binding to certain things. So there are various wet lab techniques called assays that measure all this stuff. And around the time when I was at UCLA, shotgun sequencing became popular, so the short reads were able to be read by these Illumina sequencers. Professor Pellegrini has. And so most of these techniques work by doing something chemically in the wet lab and then sequencing after. And so I will talk about two of them in a little bit, one of them being a DNA-seq and another one being ChIP-seq. And so from all these assays that I just mentioned, what we get are unbelievably large amounts of data, okay? And it usually measures certain peaks, right? So where are the peaks usually tell you either where the chromatin is open or where the transcription factor binds and so on. So we have thousands and thousands of these tracks measured by these sequencers. And across the entire genome, again, 3 billion base pairs, but it's still very hard to determine what's important in this 96%, right? So the question is, how do we know what's important? And not to confuse you any further, but there's also this other data that came around during this decade that I've been here. This is whole genomes have been sequenced for many species. Okay, so because whole genome sequencing became very cheap, not only we have the whole human genome sequence, but we have almost any possible species out there sequenced. We have their genomes as well. And then later on, I will be talking about genome alignment, aligning different genomes from different species, because we're interested in conserved regions. Again, I will get back to that. For now, I just want to make a point that we have thousands of tracks of data measured by all these incredible machines and assays. And our objective is to make sense out of it. And so this Pickle Method is attempting to summarize these thousands of tracks into a very single track, a track that's very, like a single track that's for the entire human genome. And we're hoping that that's a good resource for scientific community to help discern about among more important regions of the non-exonic genome, and then genome parts. And then we also have another goal, and that is to see how well this method could detect conservation, or what are the conserved regions in the genome. Again, I do realize that some of you might not be understanding what the conservation is yet, but I will get to that as well. So for now, I'm just going to tell you about Pickle Method. And so we are basically combining these giant tracts of functional genomics data, and we are combining the data that I just showed from conserved regions. And we are using logistic regression. So we make a model from all this. We use logistic regression to train that model based on this data. And we produce a single track with a single score. Okay, so that's very important, that there is a score or a probability at every single position in the genome. And so we're not the first ones to do this. Of course, there have been attempts fairly recently actually to use or to integrate functional genomic data and to do something with it, but most of these methods are actually not doing what Pickle is doing. They have different aims, or they have different prediction targets. A lot of them do use machine learning. Some use supervised learning, some use unsupervised learning. They use very limited number of features. So when we talk about thousands of tracks, we use many more than they do. And they are also not trying to exclude these exonic regions, while our method, I want to emphasize, is really trying to focus on that 96% of non-exonic features. And so out of these methods, maybe Schreider and Kerr one comes the closest to what Pickle is trying to do, but even that one uses GWAS data and not the functional genomics data. And also it uses it at a very coarse resolution, so they don't use the entire genome, they use parts of the genome. So our project is a little bit more comprehensive. So I'm gonna go over now for the method, how it works, given that this is a machine learning computer science thesis. I'll tell you what kind of features we're actually using, and then I'll go through the rest of it. So the features we're using, like I said, a lot of methods are limited. They use like 1,000 features or less. And I told you we have thousands of tracks. So we ended up using almost 11,000 features. So that's a huge, huge amount of features. The data is derived from these techniques that I promised to talk about that show you the states of open chromatin and histone peaks. And so we actually have 5 different data sets. Transcription factor, binding sites, histone peaks, DNase peaks, also DNase footprints, which are just a better resolution of DNase. And then we have chromatin states, which is a whole other animal. So about 50% of these are features are actually wet lab raw data, and about the other 50% are computationally derived features that are actually using Chrome-AGMM that my advisor, Dr. Ernst, created. So I'll get into that. And these, where do we get this data? Well, nice thing about the past 10 years is that all data is public. So we get it from public databases. And ENCODE and Roadmap Epigenomics are 2 big consortia trying to map all this epigenomic and non-exonic genome data. So we get a lot from there. And then there's also other public data available. So we picked the set that was curated by this group in France, I believe, just so we have a variety of features. And just in case you're bored by now, I thought to put a nice picture. So I told you about the DNA being wrapped around the histone proteins. So I think we can see that there is a double helix around, and this should be probably histone protein with proteins consisting of alpha helices and beta sheets, right? And so now I'm going to tell you more about these 2 techniques. Obviously, I'm not a biologist, so I don't know very much, but CHIP stands for chromatin immunoprecipitation. Remember now we know what chromatin is, right? followed by that shotgun sequencing. And so what it tells us is where the chromatin is open, and so where the transcription factor binding sites can bind, right? So for our data, it tells us the modifications in histone proteins, particularly in tails of histone proteins, and the transcription binding sites. But this technique also can identify some other regulatory elements as well that we're not using for this method. And another one is DNA-seq. This I'm not even going to try to pronounce, but it is an enzyme, and we know from chemistry that enzyme cleaves things, or in computer science terms, it cuts things, right? So basically, the areas where this enzyme binds and it basically cuts the DNA in the areas of where the chromatin is open. Okay, so it won't cut where it's wound and inactive, but it will cut in places where the chromatin is open. So those sites are called DNase1 hypersensitive, and so this data is basically giving us the DNA peaks that we're using for the method, for the training. And now, this is the harder part for me to explain. Dr. Ernst's brainchild, ChromAGMM. So again, similar to Pickle, the idea being let's incorporate multiple data sets, multiple data tracks into a single one, into a single score, and provide something more useful out of this vast amount of data. So what I'm showing here is 9 different cell types. So there's data from 9 different cell types. And ChromAGMM as a method uses multivariate hidden Markov model to make sense out of these epigenetic marks, to basically look at the combinations or combinational patterns of chromatin marks and put them in a single track. And you can also think of those as vectors of activity for each region in the genome. And so what you're seeing here on the left-hand side is what we call emission matrix, because hidden Markov model has the transition probabilities and emission probabilities. And so here, based off of these emission probabilities, you can see various scores or various frequencies. I said this is for 9 cell types, so 9 cell types are in the columns, and the numbers, the darker the square, the higher the probability or higher the frequency. And so when put through this method, the data has 15 different states. So these 9 cell types and all kinds of data are memorized in 15 different states. And the states are hard to read, but they're basically active promoter, weak promoter, strong enhancer, and so on. Again, those regulatory elements that I talked about in the beginning. So one data set we use is the 9 cell types, 15 states. And then there are 2 other data sets we use, and I told you it's about 50% of the features used to train Pickle are from this. And so this is the one from 127 cell tissue types. And so when the hidden Markov model is applied on that one, we end up with 25 states. And so we have 2 different data sets, 25 states from 127 cell tissue types, both, but one uses just the raw experimental data, and another one uses the imputed data as well. That's learned based off of this data. And then on the bottom, I'm showing you what that looks like in UCSC Genome Browser. Right? So this data, given that this is also part of ENCODE and Roadmap Consortia, this is actually available in the Genome Browser. And so, you can see it gets very colorful with these states. So that's all about the data. And then in order to do a logistic regression, we need something called labels. And so the labels come from conserved region data sets. I've shown you a lot of data sets where you line up different genomes. Oh, apologies. And so now I'd like to show you a toy example that will hopefully explain better. So when we compare genomes among the species, we don't do it like pairwise because we have so many. We compare like hundreds of genomes together. So we line them up as close as possible, and there are methods that do that so that these nucleotides line up. And then when we talk about conservation or conserved regions, we look for columns that basically have the nucleotide, the letter, not change over species, right? So it is considered, the area is considered conserved by evolution if it hasn't changed because it was probably important, and the nature did not allow changing those nucleotides throughout species. I'm thinking maybe breeding or something like that. And there are different methods that detect this conservation. Most of them are based on certain mutation rates, right? So the lower the mutation rate, the higher the chance of the region being conserved. And then there's some newer methods that are recognizing that maybe the mutation rate is not the only thing that plays the role in this, because there could have been some events in the evolution that weren't so linear. Maybe there was something happened in between as well. So the oldest and most used method in the work that I've done is FastCons. It really is a substitution-based method, and so is this Sci-Fi Omega. GURP also uses substitutions, but they do try to add a little bit more to it. They try to look at rejected substitutions as well, and they use maximum likelihood in order to do that. And then the newest one is this pi method, pi elements, I will be referring to later. It not only looks for substitutions, it also looks for patterns of changes. So to give you a toy example of patterns, I've told you that that 3 letters or 3 base pairs or 3 nucleotides, if you will, of the DNA code for a single amino acid. But for example, amino acid lysine has 2 codes. So AAA or AA and G both code for lysine. So that kind of pattern the pi elements are trying to discern as well. So that is about the features and the labels, and now we use logistic regression to train the model. Why logistic regression? It's a good classifier to classify the data, but I promised you a single track with a single score, right? So logistic regression not only gives you a class, whether something belongs to a class or doesn't, it also gives you a score, it gives you a probability. And on top of it, it uses logistic function, which gives a very nice separation of scores so that these probabilities get fairly even. And so that was the choice of logistic regression. Now I'm going to tell you a little bit more details about the computer science part of it, which was basically mostly what I spent the year on. And so one more thing I have to tell you about the chromosomes is that human has 23 chromosomes or 23 pairs of chromosomes, but they're different sizes, right? So chromosome 1 is 5 times larger than chromosome 21. So if we sample just random locations of the genome, we're not doing it justice. So what we try to do is we try to sample proportional to the chromosome size, make the sampling a little bit more unbiased or uniform. And so, let's see. So what we do is we randomly sample 10 million positions on the genome, and we take our, almost 11,000 features, and so we log it in the matrix very quickly and very simply. If the feature is present at that sample location in the genome, we log a 1, otherwise it remains 0. But you can imagine this is a really, really big matrix. And then for labels, we use, for example, this pi elements derived by the pi method, and we just look at whether the sample position is considered conserved region by the pi elements or not. And so if it is, it belongs to one class. If it isn't, it belongs to another class. And then one important point I want to make again is that we're excluding exons, because this project is really trying to zoom in on non-exonic regions. And so we're basically black boxing exons. So if the sample position of the chromosome ends up being an exon, we simply treat it as a non-conserved region. That's how we chose to do it. Another thing, as I said, different methods considered the amount of genome being conserved, human genome being conserved over time to be different, but most literature will tell you something between 4 and 5%. That kind of makes sense. So 4 or 5% of human genome is conserved. And so in this particular pi data set, they say that, or we calculated as well, 5.5% of the genome is conserved. If you remove the exons, it ends up being 4.1%, so it actually matches. And so that is how we create a training model. And then we do some validation of this model. And so what we do is we are testing, first of all, and then validation of the model. And so what we do is we repeat this training 23 times. We always exclude one chromosome as a test chromosome. So we basically train the classifier on 22 chromosomes and then test it on the remaining one. And we do it 23 times, every time a different chromosome is being excluded. And so now that we have the training matrix ready, and of course it has to be logged in a sparse format and so on. There's a lot of computational details I'm not going to get into, and this is done on a really big cluster. We are using the package called liblinear to perform the logistic regression for us. This is the package that's scaled on this large data set. And it's also well known and it has a lot of options, but the main reason we chose that one is because it's the only one we scaled. The way the method was coded, this is the only way, this is the only one that worked. The other option would have been to use stochastic log regression that would take chunks of samples at a time and train, because it doesn't, because of the size of the problem. And then we ensemble. We've tried different ways, but the main results is that we ensembled 10 different classifiers together. So instead of having a matrix of 10 million samples, we actually ended up having 10 matrices of 1 million samples. We trained each one separately using this regression, and then we averaged the predictions of these 10 different vectors that we received. So let's say for pi elements, what I'm showing here is this column. I'm showing an average weight of these weights that are produced by the logistic regression. Obviously, I'm not showing you the entire table because the table has like almost 11,000 rows. And we performed some regularization on this just to make sure we're not overfitting. We also tried to see how many samples we needed, and turned out with all the trade-offs that 10 million samples was a really good number. We went up to even 100 million samples, but it didn't seem to make that much difference. However, it did make a very big trade-off in terms of computational time and memory. And so we also try to regularize it with the L1 and L2 regression, L1 being the one that encourages sparsity, so that favors zero weights, and there is math behind it saying that there is like an absolute value term in the distribution that it uses. We tried L2 as well, that doesn't encourage sparsity, so it favors non-zero weights. It could be close to zero, but not zero, due to the term in the distribution being a squared term as opposed to an absolute value. And the only difference we saw was pretty much computational time. L2 took like 10 to 15 hours. And the L1 took like 10 to 15 minutes to train. And of course, creating training matrix took 2 days. So we went with L1, and so there are some zero weights in this table as well. And now the table has 4 columns because we have repeated this entire process I described 4 times for different sets of labels. So we used the omega elements and the GURP elements and Pascal's elements as well. Again, because different methods detected the conservation differently, and we wanted to see how that would compare. So the result of all this, if I have for you by now, is this promise score or probability. So we took the model we got from the logistic regression, and we took these weights and applied them to every single position in a genome. And so what I'm showing here is in the genome browser, we loaded that one single track with probabilities. This is a random position of the genome on chromosome 22. So what you're looking here is this track on the bottom that are Pickle scores. And so basically the peaks, the higher the probability is the higher the score, the lower the probability, you're looking at these flat regions. And then the other track on the bottom is pi elements. So again, the conserved regions that are used as labels and that are known to be conserved according to this element. And so there are those little dotted lines. And so as you can see, our peaks basically align with the known conserved regions. So we get very robust results, and also the regions that are not detected conserved, we are also not predicting as conserved. There's also, I will get into it, there's also some regions that Pickle is predicting as conserved, but they're not considered conserved in the other way around, called false positives and false negatives, which could be good candidates for future studies in biology. And then more technical again, this is the receiver operating curve for, in particular, pi element set. This is one way of measuring how well this logistic regression model performs. And so we have false positive rate on the x-axis and true positive rate on the y-axis. And so basically, it's trying to predict whether, for each point, how well we're predicting conservation, right? How true. And so most of you do know this measure. And what's important is the area underneath this curve. That's called AUC, or area under the curve, and the value we're getting is 0.82, which is really high. So that means the model is performing reasonably well. And then, given that we've repeated a procedure for 4 different sets, we wanted to see how they compared, right? So the one I've shown you has the area under the curve 0.82, and so does the omega, when we do this for the omega elements as well. GURP plus is close, it's even higher, 0.83, and Fascons is a little lower, 0.76. For GURP, there could be an explanation that GURP detects these conserved regions as longer segments. So when something is longer, our method is a little bit less likely to miss, right? So that might be the explanation. And for Fascons, given that I've told you it's a simple rate-based substitution method and it's older, so we have kind of expected this result to be a little bit lower. We also did pairwise correlation among all the scores that the method produced genome-wide, and they all correlate with value above 0.9, which is pretty good. And then we also try to check the robustness of our model by training our model with the same set of labels and the same number of samples and the same settings and everything, just different number of random samples, and we got the correlation of 0.97. That tells that the model is robust. It doesn't matter, what samples we choose genome-wide in the way we're doing it, it should still be giving you robust predictions. And I'm going to talk about some interesting analysis that we can do, that we have these scores. So I'm going to mention 4 or 5 briefly. I can go into details some other time. One of them is just to kind of look at the distribution of the scores, right? Because I said 4 or 5% of the genome is conserved. So if you look at the histogram here, so these are our scores. Of course, they go from 0 to 1 because they're probabilities. And this is the frequency of them. And you can see that a lot of them are very low. And then some of them are high. And that makes sense because only 4 or 5% of the genome is conserved. This one kind of says the same thing. This is the cumulative distribution of the scores. So again, the scores go on the bottom, and the cumulative distribution goes on the y-axis. And then maybe more interesting analysis of the scores if we take one set of these data tracks, so the ones derived by these DNA-seq. And we look at how do scores for DNAs from DNA's experiment compare to all bases. And if you look at this inner plot, it's interesting that there is at least one DNA-seq covered with a score of 0.25 or higher. And then we also noticed that 56% of bases, so almost half of the genome, are covered by at least one DNA-seq. So that could also have some biological significance. Another biological study that's possible is to load some of these GWAS SNPs that I mentioned earlier and kind of look and see whether they fall in the conserved regions or not, because there could be interesting biological candidates that way. So what you're looking at the top is our predictions from Pickle, and what you're looking at the bottom are the SNPs, and in particular, in this particular region, 2 SNPs. And this is a very, very preliminary analysis, so it's just an idea of what could be done with this method. And another one, interesting one, is a heritability analysis. That is, I've mentioned that SNPs are linked to the equilibrium, and that makes things very complicated and has been for the past 10 years. And so this group just came up with this method very recently, and they're trying to specifically address the linkage to equilibrium by taking certain proportions and calculating a heritability score, and hence like fold enrichment. And they use chi-square distribution, having some beliefs that the higher the value, there would be a higher heritability. So what we did with their method, we took our scores and we downloaded the SNPs for BMI, body mass index. So that is considered a complex trait. So we took all the SNPs related to BMI and we portioned them in bins, our score bins, so that each bin has about 4 to 5% of BMI SNPs in it. So the intervals of these 20 bins that are in the rows are vary because, as I said, it's been proportioned to have even number of SNPs in each bin. And what's interesting here, so there are 20 bins, and the way the analysis is done is we take one bin and we run their method against the other 19 bins. And what we end up getting is this score called enrichment, fold enrichment. And the interesting finding here is that the highest scoring bin, so when the score goes up to 1, it has 8-fold enrichment. And so this is sorted by the enrichment value. And then the plot is sorted by the intervals of the scores. So this is the highest interval, and then less and less and less, and the lowest scores are on the bottom. And if you look at that, the bins with top high scores all have enrichment of 6 to 8 times, so 6 to 8-fold. So that kind of shows the analysis that the probability of something being in a conserved region is also has a high probability, or correlates with a high probability of heritable complex trait. And there's one more analysis that comes to 4 or 5. This is an attempt to test the false negatives. So I took all the low scoring regions, so where Pickle doesn't predict there is some conservation, but the pi elements say there is a conservation there, so it's a false negative. And I ran it through this gene ontology database, and what came up very significant are these olfactory receptor genes, and a very significant high value. And the literature is mixed. Olfactory receptor activity should be highly conserved, so our method should actually not be doing well. However, if you look at other literature, it says that olfactory receptors, there's like 400s of them, and they're very, very much across the species, so they're actually not conserved. So this type of analysis could just, again, suggest like further biological studies about the conservation. So I'm going to switch gears. So whoever did not like the the machine learning portion, there is a project with monkeys. So vervets, I don't know how many of you even know what vervets are. They're monkeys. They're also known as African green monkeys. And they lived in Africa, and in colonial times, in 1600s, they were brought on ships to the islands in the Caribbean, St. Kitts and Barbados in particular. And then in the then they formed, the habitat there for hundreds of years. And then some UCLA researchers went in the 70s and trapped 57 monkeys that were like scattered across various locations of these islands and brought them to UCLA. And they had them for decades at UCLA as a vervet research colony that ended up being inbred pedigree, right? Because monkeys mated with each other, and it ended up having about 1,000 members. And then that colony moved to the university in the East Coast in January 2008. So if you'd like to see the vervets, none of us gets a chance to see them. However, before they moved them, the UCLA researchers took some data, right? And so what they did, they took data from 347 individuals, they drew blood, and they also did 2 replicates from 18 individuals just to eliminate certain technical difficulties. And then they also sacrificed 12 monkeys in order to get the brain tissue. And there's 8 types of brain tissue, or 8 brain regions in this primate model. And so the challenge is now, so now we wanted to do expression studies on this data, but we had some challenges. One being the vervets are not one of the majorly studied organisms, even though they're amazing organisms to study primates, right? And to not cut human brains. We didn't have microarrays for vervets to be able to do expression studies. And then also not very much was known about gene expression in primate brain, simply because it requires cutting the brain open. And the most data we had was actually from blood. So we had 3 goals for this project. First was to evaluate whether we could use the human sequence, how compatible it would be to use the human sequence to do a vervet expression studies. And then if that's good, then characterize gene expression in these various regions of the vervet brain. And finally, the most important one, to find the genes that are expressed both in brain and in blood. That would make life much easier, so no brains would have to be cut because these genes could be just drawn from the blood, right? And so for 3 goals, we're using 3 methods. I'll go into each one in detail. So for first one is, of course, just validating whether we could use human probes. So these were Illumina Beat Studio human probes that we were using. This was before the shotgun sequencing, so these were about 50 nucleotides long probes. And so we had 22,000 some probes there, and then we also had 350,000 vervid sequences, but we did not have a vervid genome, because again, this was also before all these genomes of various species have been sequenced. Okay, so I used the method called BLAST to compare these probes to these sequences known to be in vervid brain and blood tissue, and then I kind of calculated how much they matched. I calculated frequency of the matches. So here's another pie chart, and the takeaway from this pie chart is that 88%, or so almost 90% of these human Illumina probes matched the ones in human brain, so we could actually go on with the expression studies. And so for the expression studies, the interesting thing was we were looking for the intra-individual, so for the expressions that are varying between different individuals and the expressions that are varying, gene expressions that are varying within an individual. And again, with the goal being that we want to find these genes that are expressed in blood and in brain at the same time, and also to find the potential candidates for further biological studies for what's called EQTL mapping, which I'm going to get to in a second. And as I mentioned, the second goal was just to characterize gene expression in different parts of the vervid brain. So for that, naturally, the method was clustering. And so this is the hierarchical clustering. As you can see, it's very colorful. So different gene expression, different genes are differently expressed in different parts of the brain. And what's interesting about this is that cerebellum, like one part of the brain, clusters totally separately than other gene regions, than other brain regions. And another interesting finding is that these three parts cluster together. It turns out that all three belong to the frontal cortex. So they actually, the clustering results of our expression data do have significant biological meaning. And for the third and the most important part, to find those genes that are expressed in brain and blood, we've had a lot of data processing, data analysis, and eventually it came down to the method of just finding the variation, so percent variation between brain and blood. And we went with a cutoff of 55%. And to correct for any bias, we used Spearman rank correlation. And so out of all these numbers, replicates brain data, blood data, and so on, this is an interesting number. This intersection here is 32. So out of, about 2,500 genes, I've already mentioned to you 22,000 probes, we narrowed it down to 32 transcripts or genes. And so the results are actually ended up not being 20, and 32 ended up being 29 due to some technicalities, I think. So we found 29 transcripts that are expressed in both brain and blood, and that are all these things that are important to biology. And so the paper came out with these findings, and then there was a follow-up paper in 2012 where they actually used these transcripts and did some follow-up studies for EQTLs. So I did not mention EQTLs being expression quantitative trait loci. For the computer scientists, this just means a number. Okay, so they found this one single EQTL in a brain region that is located within only 200 kilobases by doing this SNP genotype sequencing and association studies. So they basically found a number in a very small region of the genome that has a very significant biological meaning. So that's the takeaway from the follow-up study about vervids. And I'll just take a few minutes for the third project. I've spent a lot of years in this lab being interested in sensitivity analysis and parameter set estimation, but I'm not going to talk about that. I will talk about something that actually gave result, as simple as it looks. So I've mentioned mentioned that before we had all these large data, we had just several data points measured, clinically. And so Dr. DeStefano's lab was working on this dynamic system modeling, which is basically systems of ordinary differential equations model the biological functions. And for those of you who know differential equations, the solutions to differential equations are functions, right? So that's why it's called dynamic, because you can get a function that would basically show you how the system acts over time. And so this particular model was published in 2006 by Professor DeStefano's group, and then later on this web app has been coded, and the paper about this app just came out recently. It models how the thyroid hormone flows through the body, and it also allows the users to use a hormone replacement to kind of calibrate the dosage, or to see what's the body's response to a certain dose of hormone replacements. We talk about thyroid hormone. There is T3 and T4 and TSH. Those are three components. Usually hormone replacements are only T3 and T4. So this works well, but then it turned out that this model was broken for if the dose was very high. So if the patients were given a thyrotoxic dose of hormone, then the model kind of breaks down. So if you look at these dots, and this is data from 1982, there are red dots, and so for T4 hormone, the model output is a function, right? The blue thing. So the blue line connects the red dots, that's fine. But for T3, the blue line does not connect the dots. And there is a biological explanation that the body reacts differently when it's given so much hormone in order to get rid of it, right? So what we want is something that's more like a green line. We want to fix in the model that would end up looking like a green line. And so because of the biology, we were hypothesizing that maybe just using a higher order Hill function would do the trick. And so a lot of these terms in ordinary differential equations for this model use this Michaelis-Menten kinetics, which is basically a sigmoid function. So in a logistic regression, recently, logistic regression is a sigmoid function. This is also a sigmoid function. And then the Hill function is basically another sigmoid function as well. It's just raised to the power n. So my experience with parameter search and sensitive analysis, I was just saying, how about I find a better n? And so I use a lot of algorithms, and most of them don't converge and give bad results and so on. But then I ended up finding this one that was devised in 1982 and then recently modified, called Global. And that one gave me the value of n to be 1.2, not very much. And then of course it gave me values of the other parameters, because once you mess one thing up in the model, one of the parameters, then you have to fit the other parameters as well. And so once I plug that in to the model, it actually does connect the dots. And so if you look at the T3, which is the bottom one, now it actually does connect the dots. And we have two data sets, the blue and the red. And in both cases, the model performs reasonably well, and the error bars are not that high. And so now, I don't know the time, but hopefully it's not too long. I'd like to give acknowledgments. I think this is my defense, so I want to spend a little more time on this. First of all, this is Zylab in 2007 when I joined. Most of these people are very successful PhDs, professors, and so on. And I'd like to thank my advisor and chair, Dr. Eskin, my hero, who taught me into all this. And then during those years, I was actually working with Dr. Freimer and Dr. Jasinska. She's actually here, so she did the Vervet project. And so these were my beginning years at UCLA before I took a break. And then my third project, my other hero, my new advisor, Dr. Ernst, is here, and here is his current bioinformatics lab. And I'd like to thank you all for being here, and thank you all for this extra year that I stayed in school, and that I got to know you, and I got to learn so much, and I got to learn epigenetics and so on. And especially want to thank Shalvi and Adriana for making some of these plots for the score analysis. And I'd like to thank Pete for teaching me so much and giving me so many useful suggestions. Now, another person I want to thank is my undergrad advisor, Dr. Kelly DeViewer. She doesn't even know I am doing this, but she was the one who made programming fun. And she was also the one who talked me into attending the ACM programming competition. So this was us in 2003, I believe. Our team was named Fire Ants. And if you can imagine, we didn't do so well at the regional competition. And then turns out that our professor here, David Smallberg, is training the UCLA team for the Olympics. And I would like to thank Professor Smallberg. I've spent 6 years TAing while here at UCLA, and at least half of that time were his programming courses. So he was a mentor to me in a way, and he also taught me a lot of programming. So I think this team would have done much better right now. So that's it. Again, thank you for the heroes, Dr. Eskin and Dr. Ernst, and Dr. Jasinska for the project. Thank you for the committee. And then before we go to like a real question and answer session, I want to answer one question, if you allow me first. And that is the question that I've been, asked many, many times in almost 2 decades in computer science, and math and STEM fields. So it's always why computer science isn't it hard? And then another one, why a woman? As a woman, why are you in computer science? I mean, not to say, that's like the nicest way to say it. And so it just so happened that I lived 2 blocks away from this house. And this house has this plaque on it that you can't read on the slide, but you can actually read if you look at the picture. And on my way to and from high school for 4 years, I was passing by this plaque. And so this is a first wife of Albert Einstein. Her name is Mileva Maric, and she was living in this house prior to going to school in Zurich for mathematics, and where she met Albert Einstein. And they got married, they were living in Europe, but in actually 1905 and 1907, they were living in that house. So the story I was told, and mostly it's factual, it can be verified, but the story I was told is that she was a talented mathematician. She was his collaborator, which is also indicated in the plaque, and she did a lot of math for him. And on top of it, she did receive the Nobel Prize money after he received it, even though they were divorced at the time. And so also another fact is that he did his best work while being married to her. And so to me, passing by this plaque, it was always a sign that women can do math. And it took me until the 4th year of high school to actually learn the theory of relativity, because I had to take so many advanced math courses to get to that level in physics to be able to understand it. And so the answer to the question I posed, or that's been posed to me, is not that I've been encouraged to go in the STEM fields. I've always liked math and computer science. I was good at it. It's just that I have never been discouraged. And so that's the message I want to pass, because sometimes I don't really like to go and say we need more women in computer science and so on. I'm a big champion but at the same time, I think a lot of times it's just to not discourage those who do want to do it. And I'd like to thank you all for coming here. The list would be too long. It would be like a Oscar speech, right? So I will save that for my long acknowledgement page in the thesis. And I would also like to thank my parents, who, this is a 3-year-old picture. I have not seen my parents in 3 years because of the sacrifices I made in order to finish this PhD. And they're very, very sad that they're not present. So I decided to put them here.
Thank you. Any questions? I guess we can start with the committee first. Okay, let's see if you have any questions. Mateo? Huh? No. No question. Mateo?
So how do you, so turning to the first thing you talked about, the pickle, where do you see the main application of the approach? Like what was the motivation?
So like I said, so the question was, what's the main motivation of the pickle? And I like that you use the word. So the main is to try to combine all these multiple functional genomics tracks into a single one and give a certain probability and a tool to a scientific community to prioritize certain locations. And in particular, it focuses on these conserved regions in non-exonic things, but I've shown several ways, other ways that could be used for analysis that would eventually lead to some more contribution into understanding the complex traits and diseases.
So see it as a way to prioritize regions
Yes.
of the genome that are more likely to be functional.
Correct. Similar to what the Chromagem, which you're probably familiar with, does. It combines a lot of things and gives you 15 states. We give a single score for each position genome-wide. It can't be that it was that clear. Come on.
Jason, I think I passed through. Okay. So, I have actually another question about your first project. So, what kind of data sets do you see that are like? Is there a type of data you think that if it was collected, it would make the signal a lot stronger?
So what would be the data that would make the signal more stronger?
Or like, do you see other data types of data becoming available that would make the signal stronger? Like high C or other things?
I honestly don't at this point, because we have collected so many classes of them. I know that RNA data we did not include because the RNA is already well associated with exonic. So with high C data, I'm not as familiar. So I'm not sure what else would be. Anything that would show the areas of open chromatin, I think would be the type of assay. We did use FairSeq for part of it as well, because it also shows the open chromatin, because I think most features are done with related to open chromatin in places where transcription factors can bind.
And then how tissue specific do you think is the?
So we didn't. There was another classifier that was trying to be sour tissue specific. The one that we did, we kind of combined everything together into one classifier. So chromatin states have differentiated between the tissues, but this particular method hasn't.
And so do you see that if you have a lot more data for a specific tissue, you get some benefit from building a tissue specific score?
Yes, I do. I think that would be a wonderful extension. That, if we have, instead of thousands of tracks, like we had like gigabytes of data, now we have terabytes or petabytes of data. I think that if we have, thousands of tracks that are just simply, let's say, stem cell specific, then we could just trained the model using those. And we made the model flexible so we could add more data to it. My particular interest would be back to the Vervet project that I started with. I'm very interested in these brain regions and in traits or diseases like schizophrenia, bipolar, things that are very hard to discern.
Are there any more questions from outside the community? Okay, so let's thank Olivera one more time.